So just to piggyback on what Hamza was saying about the metabolism,
Hmm.
I think that another drawback of that, just thinking back to microbiology, when we're doing kind of like those find out what your bacteria is, like, everybody in lab would be like, do you think this is a positive or a negative result? So it's
Yeah.
like very subjective whether what you're seeing is what you're seeing or not.
Yeah, methodology matters, right? And so with all of these methods, you have to do them properly and correctly in order to get them to give you the answers that are meaningful, right? So that's a really good point, Cecilia. What are some things we do, or what's a major approach we use to make sure that we're getting an accurate result, say with a metabolic test? Like if I ran a class and I said classify this unknown organism using this test, what would you ask for to make sure that your results were reliable?
Well, you always need like a negative control, right?
A negative control, but I actually would prefer to see a positive control as well,
Right.
right?
Mhm.
Like if I know E. coli does this metabolism, right? And I want to know if my unknown organism is E. coli, I better have E. coli so I can test it using the
Yeah.
same batch of medium under the same conditions and with my hands, right?
Right.
Genetic sequencing. I'd want to have negative and positive controls for that as well to make sure that. So, all of these methods would need to be properly controlled if you are going to really use them as effectively as possible. So, that's a great point. Thank you, Cecilia. All right. Any other methods that any of the groups thought of? We've covered most of them. All right. Okay, well, that got us off to a good start. So I think we've got a sense. Are there any questions about that? We're going to talk a lot about methods, particularly like molecular methods later in the course, but I just like us to think about this right now in terms of classification. And so I do have some slides here that just basically highlight kind of some of the things we've talked about. One of the things that wasn't mentioned was the presence or absence of organelles, right? Who has organelles again?
Eukaryotes.
Eukaryotes have true organelles, although as you can see from this bacterium, there are inclusions and there are elements like the nucleoid. We don't have a nucleus, but we do have DNA, right? So there are structures, but it's very fundamentally different from eukaryotes. Metabolism was mentioned, and I just like to highlight that microbes are much more diverse than eukaryotes, right? As was mentioned, as Hamza pointed out. So plants are photosynthetic, and animals are heterotrophic, right? We have all of that and more in the microbial realm, and we're going to talk a lot this semester about the diversity of metabolisms. Pretty much you name it, and there's something out there that does it in the microbial realm. As was already mentioned, genetic sequencing is a powerful tool. One thing you can do is single gene sequencing, and we'll talk about this, versus sequencing whole genomes, or as was mentioned, sequencing whole metagenomes, right? And we'll talk about the difference and distinctions there, kind of along the lines of what Victor mentioned. Pigmentation can be important, particularly in photosynthetic organisms or others that are pigmented. So things like chlorophyll molecules, of course, are well known, but we also have carotenoid molecules. These are the things that the leaves, when they look red or yellow after the chlorophyll degrades in the fall, these are the pigments that are the reddish and orange ones. So when we talk about metabolic classifications of life, we need to think about what the carbon source is. I think Victor, or no, maybe Hamza was the one who said energy source, right? Um, so, and then where you get electrons from. And when we have our section on metabolism later in the semester, and of course, if you're taking bacterial physiology and bacteria biochemistry, you talk about these as well, right? We don't go into all the nitty gritty details of making you memorize, proton pumps and all that kind of stuff. But we do need to think about what it means for the ecology or lifestyle of where you get your carbon, your energy, and your reducing power from. All right, so here's just a quick quiz, a little poll. So we'll use these also to get everybody engaged. So a bacterium that decomposes plant organic matter would be described as which of the following 5? So let me set up a new poll. All right, and we'll launch it. Oops. Okay, what's happening? I don't want to do that. Okay. All right. Sorry about that. So you're just going to use multiple choice and you'll answer 1 through 5. Let's see. A, B, C, E. Okay. So I'm not typing the, okay. I'm not typing the question because you have that on the slide, but just go ahead and, all right. Go ahead and log your answers. All right, and you're not graded on whether you get this right, so just go ahead and take a guess if you're a little uncertain. And we'll talk about the answer. All right. So we had the majority of people put D, chemoheterotrophic, but that was only 54%. So almost half of you put one of the other answers. So somebody who put chemoautotrophic, why did you put that?
Yeah, I'll go. Yeah, I chose D because I saw that it decomposes plant organic matter. So it's not necessarily like, taking energy from the sun for like, let's say carbon source. And I know that auto kind of means that it's like it's making its own things. So I kind of just guess like hetero is like, it's the opposite. So it's not making its own things per se on the same level as, let's say, a photosynthetic bacteria.
So actually chemoautotrophic is what you would say for photosynthetic bacteria, where they are fixing their own CO2. So it's a little tricky, I think, because I put plants here. So the plant is chemoautotrophic, but the bacteria that decomposes it would not be autotrophic because it's using the carbon the plant already fixed from the atmosphere. Does that make sense? So actually it's not an autotroph, it's a heterotroph. And so then the best answer would be, is it A or D? And most of you put D, and so that is the correct answer. So I think most of you that put A probably got tripped up by the fact that it said plant material. And so ultimately it came from the sun and from, Rubisco and carbon fixation. But for the bacterium themselves, we would classify them as chemo, getting their energy chemically from the dead rotting material, but heterotrophic, using carbon fixed by somebody else, not themselves. Does that make sense? Does everybody see why that distinction is there? Yes? You guys can give me thumbs up. Okay, cool. All right, any questions about that? Okay. Okay, so which bacteria would likely be in this category then? So let's relaunch the poll. And please don't put E this time. Put A through A or B, C, or D. Would it be a cyanobacterium? Would it be a gut coliform, a sulfur vent bacterium, like in the deep sea? You guys have seen those, I'm sure, or none of the above. Would the plant decomposer fall into any of these categories, or would it have to be none? All right. Go ahead and finalize your vote. Okay, last few of you. So please do fill it out because you get participation for participating in the polls. So go ahead and give me an answer. Awesome. Okay. So it looks pretty pretty evenly split between B and D. So why is it not a cyanobacterium? Oops, gosh, something keeps going wrong with this. Here we go. Why is it not the cyanobacterium?
Because it uses CO2.
Right, so cyanos use CO2, and in this case, they're not doing that, right?
Correct.
Okay, so a gut coliform, right? So a coliform bacteria like E. coli, right, lives in your gut. Do you think a coliform bacteria could potentially break down plant material?
Yes.
It'd be behaving as a chemoheterotroph in that case, right? Yeah. So I can see why people keeps jumping. I can see why folks would put none of the above, because we don't normally think of gut coliforms as being plant decomposers. But when you eat vegetable matter, right, your intestinal chemoheterotrophs are actually breaking down that lettuce that you ate or that whatever. Not well, but they do. So there are other plant decomposers that are much more effective. And so putting D is actually not the worst answer here either, because it could be something different. But gut coliform is the best answer. That's the one that I was going for, but I can see why you'd put D. All right. Okay. All right. So when we look at genes or genomes, we need to think about it being in pure culture or in the environment. And we're going to talk a lot about that later during the course. But one of the other things we need to think about is the impact of horizontal gene transfer or lateral transfer. So when you guys learned about making the tree of life in like general biology, do you remember what gene or genes they talked about were used for that? When you saw a typical tree of life, did they use entire genomes for that, do you think, or were they using single genes?
Single genes?
And do you remember what gene was?
RNA?
Okay, so it's the ribosomal RNA, right?
Yep.
Okay, so ribosomal RNA genes were early, and we're going to touch on that in another slide. But we do have to be aware that when you're sequencing, and ribosomal RNA is pretty good because it's less subject to this lateral gene transfer than some others are. But if you sequence just a single gene, or even the entire genome, it can be challenging to know, is that gene ancestral to that group, like that group of bacteria, say, has always had that gene? Or is it something that was moved over on a plasmid or some other, like by a virus, some other kind of horizontal mechanism? And so that's one of the challenges that we'll also discuss a bit this semester. All right, so that is what I wanted to cover in terms of classification or taxonomy, just to kind of have us all be on the same playing field with that. Are there any questions about anything we talked about there? All right, so this is just a kind of fanciful image. It's quite old at this point, but it doesn't really matter. This was taken from Scientific American. but it provides you with kind of a glimpse into what we think the Earth was like many, many billions of years ago, right? I know it's hard for us as humans, you know, we live maybe 100 years if we're lucky, right? To think in terms of even thousands of years, much less billions of years, which is 1,000 million years, right? But the geologists tell us that the Earth formed over 4.5 billion years ago, right? And of course, there wasn't life right at the beginning, and it wasn't a very hospitable environment. But it's remarkable how quickly life is thought to have evolved pretty quickly thereafter. But one thing we need to think about is that the conditions on the early Earth were very different than they are right now. Okay, can anybody think of anything that would have been different, let's say 3.8 billion years ago when we think life may have evolved?
The environment would have been anoxic.
Okay, so there wasn't oxygen, right? Because where does the oxygen in our atmosphere come from primarily?
Cyanobacteria.
Cyanobacteria and of course plants, right? And that didn't come for billions of years later, right? Okay, so it would have been anoxic. What else? And you can use this image to kind of give you inspiration if you want. Say again, I'm sorry. Can you say
The
that again?
temperature?
Temperature, what about it?
Yeah, it probably was a lot hotter or a lot colder.
Yes.
Since there wasn't a lot of cycling of oxygen stuff.
Well, so oxygen doesn't really impact the temperature, but yes, it's thought that the Earth was much hotter at that point, and it's sort of cooled. There was much higher volcanic activity, as you can see depicted here. And so the early Earth was likely very much warmer. The oxygen in the atmosphere would impact what Jenny put in the chat, which is absolutely correct, is that there's higher ultraviolet radiation levels. Okay, so why would that be? Why does oxygen matter for UV?
Presence of ozone, or lack of it, rather.
Yeah, so if you don't have O2, you don't have O3 in appreciable amounts either. And as I hope you all are well aware, the ozone, the O3 layer up in the atmosphere, in the stratosphere, protects us from ultraviolet radiation. Anything else that anybody can think about? These are all great answers. All right. So yeah, so in terms of what we would consider, and remember, this is from a human standpoint, it would have been quite hostile. So in the early solar system, when the Earth was forming, there would have been collisions that go on. And in fact, the general scientific consensus is that our moon was formed through a massive collision that must have occurred billions of years ago, which actually ejected the moon, what we now know as our moon, off the surface. So the moon is made of Earth rocks, but when that collision occurred, you can imagine how big an explosion that would cause to have to drive an entire moon's worth of rock off of the planet. So when these bombardments were happening, even smaller bombardments would have also sterilized the planet at that point. So life could not exist when that moon event happened. So we talked about temperatures, and things really didn't cool down probably for hundreds of millions of years. We talked about oxygen, we talked about UV. The gases in the air would have been very different as well. So there was more CO2, and probably higher levels of methane. Now, I hope you're aware that we have lots of N2 gas, right? Like 79% of the atmosphere right now is nitrogen gas. But the composition overall of the atmosphere would have been very different. And you would have had traces of some things. And these traces actually would have been important because they would have provided energy. So if you see an H, remember, we need to think, oh, there's electrons associated with that. And so hydrogen gas and things like hydrogen sulfide and iron sulfur are good electron donors for metabolism. And so when life did evolve, this may have played an important role. So there's quite, I want to preface the whole discussion of the origins of life with the fact that we don't know how life evolved, right? We can speculate, we have evidence, and I'm going to present some of that here in this slide. But the reality is we don't know how it happened. We think we have a pretty good handle on when it happened, and that's here. Over 3.8 billion years ago, there are isotopic remnants in rocks. And that's shown here. This is a study that came out decades ago now. There have been follow-up works that have confirmed this, though. If you go to some parts of the world, in Greenland and Australia, there's actually really old rocks. So much of the Earth, because of plate tectonics, has been subducted and taken down back into the mantle and melted and metamorphosed and all those things that you've learned if you took a general geology course. So most of the Earth's surface is actually relatively young, and that's how we end up with places like California, where you've got plates crashing together and mountains forming and earthquakes happening and volcanoes forming. But in some really remote parts of Central Australia